Hello everyone. So today's lecture's topic is gingival clavicular fluid, GCF. It is also known as a window to periodontal disease. Why it is said so, we will see in the lecture. So the contents are introduction and history, formation, methods of collection, functions, composition, humoral immune response, GCF flow, GCF as in diagnostic test or risk predictor for periodontal disease, clinical significance. So we will be starting with introduction and history. So GCF studies extended over a period of about 50 years. Presence of GCF has been known since 19th century. It is either a transudate or an exudate. This is a question. The main route of the gingival fluid diffusion is through the relatively wide intercellular spaces of junctional epithelium and then into the sulcus. Little or no fluid can be collected and in strictly normal gingiva. So here we can easily see the fluid which has B for bacteria, D for degradation products, E for epithelium. This we can easily demarcate where is epithelium. Uh, Actually, wait for a second. So here we can easily see that this is the epithelium. Then the fibroblasts which are present over here. Then in this fluid there are neutrophils. Then there are osteoclasts. Fine. And then we have serum. This is serum. Serum which travels from this side to that side. Osteoclasts are present over here. You can easily demarcate. So it is a complex mixture of substances derived from serum, leukocytes, connective tissue, epithelium, inflammatory cells, and microbial flora inhibiting the gingival margin or the sulcus, the pocket. So, in earlier 1950s, panel research of Jens Wehrhach, it was based on the anatomy of the sulcus and its transformation into pocket during course of periodontitis. In late 1950s or early 1960s, Brill et al. Did a research which was based on physiology of GCM formation and its composition. Louis et al. Introduced GCF as an indicator of periodontal disease. Egelberg, Egelberg continued to analyze gingiva and on into gingival blood vessels and their permeability as related to GCF flow. So Edelberg continued to analyze this GCF and on dendrogingival blood vessels and their permeability as related to GCF flow. In 1970, Suta, Bang, and Chinasumi did an experiment to include the presence and functions of proteins, especially enzymes in GCF. Olison, Golub, and YouTube, they did work on collagenase and elastase in GCF, which are derived from neutrophils, and their activity correlates with gingival inflammation and pocket depth. Edstrom et al. Studied migration of neutrophils and its function in GCF. Alfano hypothesis: GCF isn't transitive, but in course of gingivitis and periodontitis, it transforms into inflammatory exudates. This hypothesis was given by Alfano, Dilemma, and Venn Diab. They did studies on cells of GCF and find balance between protective and destructive roles of neutrophils derived post defense. LA and Cox, they studied on properties of oral bacterial enzymes and their value in diagnosis of periodontal disease. Ebersole studied antibodies in GCF and its role in maintaining local and systemic hemostasis. Gianabel et al. Studied bone-specific markers like collagen, telopeptide, osteocalcin in periodontal bone resorption. Champion et al. Studied inflammatory mediator levels in periodontal disease. So you should be knowing that who gave the hypothesis that GCF is an transutate or an inflammatory exudate. So the answer is Alfano. This is important. Now the formation. Is GCF an inflammatory exudate? So the initial investigators that are that were Brill and Brasse in 1958 and in Eaglesburg. So what they hypothesized is an increase in vascular permeability of the vessels underlying junctional and circular epithelium. Some irritation, it can be chemical or mechanical, which is necessary to induce the production. So it is considered as a pathologic phenomenon. So author Brill and Cressy in 1978, their samples were dogs. They introduced filter paper into the dog's sulcus, which was previously injected with fluorescence given by through IM within 3 mm. Fluorescent material was recovered in the paper strips. What were the results? The passage of fluid from the bloodstream through the tissue and exiting by the gingival cells. So they did another experiment in which samples were dogs only. Stimulation of gingiva by toothbrushing, chewing, IV injection of histamine, development of inflammation. So the results were markedly increased flow of gingival fluid. So now what was the experiment of Eagleburg? He experimented in dogs. What he did was suspension of carbon particles of known particle size. He injected them IV and then dog killed and the gingival tissue was examined histologically. So the control that are healthy specimens. Carbon particles remain within capillaries and small venules. Presence of acute inflammation, particles could be seen in intercellular spaces because of increased vascular permeability. So now what was the theory of Alfano? Clinically healthy gingival crevice harbors bacterial plaque. Accumulation of high molecular weight molecules permeate intercellular regions of epithelium, but limited by basement membrane. So accumulation of high molecular weight molecules, produce an osmotic gradient, induce a flow of interstitial fluid from connective tissue to the gingival sulcus. So, phosphate buffered saline, that is 10 mg into 1 ml of homologous serum albumin, results in 100% increase in the volume of GCF. Phosphate buffered saline alone did not enhance the volume of GCF produced. So, then what was Paschal's hypothesis? Cross section of two capillaries, longitudinal section of lymph vessels. It showed absence of inflammation, that is low vascular permeability, low permeability of the basement membrane, low GCF flow, high percentage uptake of lymph vessels. Macromolecules of plaque osmotic gradient, increased vascular permeability, basement membrane changes, increased passage of fluid into the tissue, and increased GCF production. So the methods of collection was number one, absorbent filter paper strips, either intraclavicular, that was press intracellular circular technique, or extraclavicular, that was Louis and Hall Pedersen extracircular technique. The techniques can be easily understood from the diagrams. Please go through them. So now what was the advantages? Simple, quick, and easy to use. View directly. Quantitative assessment applied to individual sites. Compatible with subsequent chemical analysis. And by using periotrone, it was a minimum evaporation would have happened. Disadvantages: dislocation of paper strip, disturbing the integrity of the marginal tissue, chances of contamination. In case of evaporation of sample, it has to be repeated many times. If periotrone is used, daily check on the reading accuracy should be performed. Care to be taken. Insert strip into machine in a standardized position for correct reading. Then the methods of estimating the volume collected. Number one, linear measurement. Distance the fluid had migrated up the strip or assessing the area of filter paper wetted by GCL. Second, staining. More accurate value. Stain with an alcoholic solution of thin hydrate taken in 0.2%. Blue or purple color stains protein. Measured planimetrically on an enlarged photograph, calibrated magnifying glass microscope with calipers on transparent scale. By weighting the strip weight for collection within a sealed microcentrifugation plastic tube and weighted immediately after collection. So this was the comparison of protein concentration on the y-axis and tissue fluid, which included serum, lymph, GCF, inflammatory fluids. So the fourth is an electronic method. It is rapid, latest, sorry, rapid, latest, and accurate determination. Fluid collected on a blotter, that is paper, employing an electric transducer, that is periotrone. Wetness of paper strip affects flow of electric current and gives digital readout. Dry strip placed between jaws, capacitance, uh, that shows capacitance, sorry, translated by electrical circuit, it registers 0 on digital readout. So wet drip, that That means increased capacitance and increased value in the readout. So, three models of periodrome, that is 600, 6,000, and 8,000 were taken. Limitations unable to measure less than 1 micron depth. Then, which Himasuni in 1983, Himasuni in 1983 used 1.5 mm white paper strip inserted 1 mm within sulcus, slightly inflamed gingiva, zero point 0.1 mg of GCF in 33 minutes was gained. Chalakombe in 1980, isotope dilution method was used. He used isotope dilution method, mean GCF volume in proximal spaces of mobility 0.43 to 1.56 micron liter and interior T 0.24 to 0.43. So basically 0.5 to 2.5 micron liter fluid per day was collected. Then 2-gram fluorescent given systemically. Okay. In this systemically to patient 2 hours prior to collection, examined under UV light, used to determine presence or absence rather than quantity. Fluorescent labeling is 100 times more sensitive than anhydride for staining. This has to be remembered that fluorescent labeling is 100 times more sensitive than anhydride for staining protein. Now, what are the disadvantages of staining technique? Not easily applied at the chair site. Protein staining prevents further lab investigation of other components. Delay in measuring result in increased variation in the reported volume as a result of evaporation. So, Winston technique, pre-weight twisted threads which were placed in gingival crevice. So, amount of fluid collected was estimated by weighing the sample thread. Then capillary tubing or micropipettes, which was introduced by Krasik and Eagleberg. Isolation and drying of a site. Capillary tube of a known diameter and length inserted into entrance of sulcus. GCF migrates through capillary action. Content is later centrifuged and analyzed or forced out with jet air or passing a large amount of diluting solution through capillaries. So now Winston technique. What was Winston technique? Winston used pre-weighted twisted threads which was placed in gingival crevice. So amount of fluid collected estimated by the amount of fluid collected was estimated by weighing the sample thread. This was very easy method. Then came the capillary tubing or micropipettes, which was Kressel and Eagleberg's method. Isolation and drying of a site was step. The first and foremost step was isolation and drying of a site. Then capillary tube of a known diameter and length was inserted into entrance of the sulcus. Fine. It was inserted into the entrance of the sulcus. Then GCF migrates through capillary action. Content is later centrifugized and analyzed or forced out with jet of air or passing a large amount of diluted solution through the capillaries. So advantages: provide an undiluted sample, accurate assessment of volume. Disadvantages: difficult to collect in short period unless inflamed sites are there which contain large volume of GCF. Collection times from an individual site may exceed 30 minutes. Samples from healthy crevices was impossible to obtain. Difficult to hold tube for such lengthy periods, plus ensuring a dramatic collection, difficulty in complete removal of sample from tube. If collection of fluid is difficult because viscosity of the fluid makes aspiration through a pipette very difficult. So these were the disadvantages of the experiment. Crevicular washings to gingival crevice is perfused with an isotonic solution such as Hank's balanced salt solution of fixed volume. Collected fluid represents a dilution of GCF and contains both cells and soluble constituents such as plasma protein. So in this, we can easily understand this through the following diagram. Okay. Then came the Stocksy and Linear method. It was the simplest method. Installation and re-aspiration of 10 micron of solution at the interdental papilla. Two injection needles fitted one within the other. So inside, two needles were used, right? One was placed inside and one was fitted inside of the another one. So inside needle, which was ejection needle, was placed at the bottom of the pocket, and outside, that is the collecting needle, was placed at the gingival margin. Collection needle is drained into a sample tube by continuous suction. So this process was repeated 12 times, which allows thorough mixing of the transport solution and GCF applied either to individual interdental units or to multiple units, which were then pulled. So, what were the advantages? Useful in clinical normal gingiva, useful also. Useful for studying number and functional state of cells and bacteria from crevicular area. Total count and differential count of lymphocytes can be applied to individual sites, group of sites in healthy or inflamed. Disadvantage: all fluid may not be recovered during aspiration and re-aspiration procedure. Thus, accurate quantification of volume composition is not possible as the precise dilution factor cannot be determined. So, then came the Tokamoli and Oppenheim method. It is a more complicated method. It uses hard acrylic plate covering the maxilla with soft borders and grooves connected to four collection tubes. The tissues were irrigated for 15 minutes with the saline solution using a periostatic pump, and diluted GCF was obtained. Advantages: useful for longitudinal studies, does not disturb marginal tissue integrity, least contamination, constituents of various enzymes and number of cells, that is, neutrophils and epithelial cells, were detected. So now what were the disadvantages? So disadvantages were complicated production of acrylic customized stents and technically demanding, only applied to maxillary arch because of the difficulties of producing a technically satisfactory appliances for mandibular arch, dilution of trabecular fluid. It was a complex procedure, and GCF from individual sites cannot be analyzed. So other methods were plastic strips, platinum loops, which were placed along the long axis of tooth, inserted into sulcus, and pressure is applied. So now problems with the GCF collection was: number one, contamination. Major source was blood, saliva, and plaque. So sampling time was from early literature from periodon 5 seconds, and through alternative approach it was 20 to 30 minutes. Prolonged collection time was nature of sample is likely to change. We all know that. Volume determination, significant problem was evaporation. Then total volume collected are usually less than 1 liter. Recovery from strips, enwrappment within or binding of proteins to filtered paper. So there will be another part of this lecture. In this lecture, we will just finish off. So please remember what we have studied. We have completed the introduction and history of GCF. We have completed formation process of GCF, and we have seen different methods of collection of GCF. In future lecture, we will see functions of GCF, composition, humoral immune response, GCF flow, GCF as a diagnostic test, uh, what are the risk predictors of periodontal disease, how GCF is related to periodontal disease, what is the role of GCF in periodontal disease, and the clinical significance. Thank you so much. Please be there for lecture, the second part of this lecture. Thank you.